My name is Deana Crouser, and today I'm going to be talking about navigating change. I'm going to start with a little background. I was raised in Miami, Florida, and I am a first-generation Cuban American. I moved to Seattle, Washington in 2016 with an associate's degree under my belt, and by July of 2019, at the ripe age of 29, I was graduating with my Bachelor's of Science from the University of Washington School of Oceanography. And my specialty, is in biological oceanography. But more specifically, I was a zooplankton taxonomist. I spent a year learning to identify hundreds of different species of zooplankton, from copepods like plankton from SpongeBob SquarePants to different types of jellyfish, shrimp, larval squid, and even marine worms. Zooplankton taxonomy can be considered a heritage skill or an endangered expertise, as few new specialists are entering the field despite its enduring importance. Mastery in this area is essential for keeping historical sampling methods alive and continuing long-term observations, which are invaluable for ecosystem research. This expertise plays a crucial role in tracking and understanding long-term changes over time, even as interest in developing this skill declines. Shortly after graduation, I accepted a position with Linker, a premier science, engineering, and technology contracting company, which supports some of the nation's most important missions. And my mission was to support NOAA's Alaska Fisheries Science Center as a zooplankton ecologist, so quite a long ways from Miami. Zooplankton are drifting animals found in nearly every body of water around the world. They can survive and thrive in some of the world's most extreme environments, from the deepest parts of the ocean, like the Mariana Trench, to the polar seas of the Arctic and Antarctic regions. They've been observed near hydrothermal vents, where temperatures can reach up to 400 degrees Celsius, that's 725 degrees Fahrenheit, as well as hypersaline lakes like the Dead Sea and highly acidic lakes found near volcanic regions. There are hundreds of different types of zooplankton, and a few are imaged here. From left to right, we have jellyfish, euphausids, amphipods, sea angels, which are a type of sea slug, and sea butterflies, which are a type of sea snail that are eaten by the sea slugs. But my favorite type of zooplankton are copepods, like this one photographed here, species Calanus marshalli. And they're my favorite for a few reasons, like the fact that they're fun to identify, and each species has a unique personality. Some can be described as ferocious, while others can be described as elegant. But the biggest reason they're my favorite is because copepods are the vast majority of the zooplankton biomass. Copepods are at the heart of the marine food webs, linking energy produced by phytoplankton to higher trophic levels of the ecosystem. They play a key role in providing vital nutrition to fish during their early larval stages, when they're most vulnerable, and increasing their survival rates. They are an essential food source for all commercially important fish species, as well as seabirds and marine mammals. By transferring energy up the food web, they help power the entire marine ecosystem. And since copepods aren't harvested directly, changes in their population can reflect shifts in the environment. Now, I know that we are all intimately aware that the Bering Sea system is warming. And these changes have the potential to trigger notable shifts in Alaska's large marine ecosystems. I want to introduce you to a little ecological theory. And as I continue this presentation, I want you to try and keep these three main concepts in mind. Now, this is real-life training for becoming an ecologist yourself, and you will be quizzed, so don't say I didn't warn you. All right, here we go. According to ecological theory, we can expect three main responses to warming. The first is a change in species phenology. Species phenology is the timing of life cycle events like growth, maturation, and reproduction. So I'm going to use my copepods as an example. Some species undergo diapause just about the time winter rolls around. And it's like a form of hibernation. Just before they reach their reproductive stage, they bulk up on as much food as they can, and they sink down to the depths on the outer shelf to avoid predation, and they ride out the dark, cold winter when there isn't enough light to support their food source, which is phytoplankton. They receive a biological cue, which could be temperature, it could be light availability, I can't say for sure, telling them when it's time to come out of diapause. They'll then molt to their final adult stage and lay their eggs just in time for their offspring to capitalize on the spring phytoplankton bloom that occurs, giving them the best chances at survival during their early life stages, which is pretty cool. But each one of those steps, the timing of when that happens, is their phenology. Okay, so the second response to warming is a shift in species distribution, as warming leads some species to shift their ranges. This phenomenon is known as borealization and involves species migrating northward to seek their preferred temperatures. One study, which focuses on phytoplankton size, observes that smaller species were found near the equator where water temperatures were warmest. And as we moved closer to the poles, we saw the overall size of phytoplankton increase. So if we have a shift of smaller, warm water species northward, how are their respective predators going to respond? Such shifts can alter local food webs and potentially impact ecosystem stability. And that's just a little bit of food for thought. The third ecological response to warming, which my research has focused on, is a decline in body size. Now, for copepods, a decline in body size can have significant implications for the entire food web, considering their role in the transfer of energy to the upper trophic levels. And during my time at the Alaska Fisheries Science Center, I was tasked with establishing a baseline for copepod size in the past conditions in order to predict the future and see what's going to happen as our ecosystems change. So I'd like to take a moment to introduce you to NOAA's research vessel, the Oscar Dyson, whose home port is located in Kodiak, Alaska. Over the past 5 years, she has been my home away from home. I have made some lasting friendships along the way, from the many scientists that collaborate with our center to the merchant mariners that make our work possible, helping us deploy and recover a wide variety of scientific instruments, to the NOAA Corps who navigate this vessel with knowledge and precision that I can only admire. The Oscar Dyson is a 208-foot stern trawler capable of conducting both fishing and oceanographic research with 6 onboard laboratories, so it's a scientist dream. She's also akin to a floating weather station. With the call sign WTEP, she's been collecting and sharing navigation, weather, and oceanographic data since 2007. And I had the pleasure of first setting sail on her during a global pandemic in the spring of 2021 for our program's Spring Mooring Cruise. Twice a year in the spring and fall, our program sets out to turn around various moorings strategically located across Alaska's large marine ecosystems. They're biophysical instruments that collect continuous biological and oceanographic data, allowing scientists to observe changing ocean conditions in near real time. These moorings provide measurements of temperature, salinity, chlorophyll, CO2, pH, as well as acoustic data, and that's just to name a few of their capabilities. But deployment and recovery of these scientific instruments are no small feat. Science and crew race against time to recover these moorings before the ice edge arrives, which would make them inaccessible to vessels like the Oscar Dyson. They brave rough seas in an effort to preserve long-running time series which provide the pulse of the Bering Sea. The most fondly recognized mooring site is M2, which is located in the southeastern Bering Sea. Affectionately referred to as Peggy, after Peggy Dyson, the voice of the North Pacific. Starting in 1965, during the booming expansion of the king crab fishery, Peggy Dyson provided the weather forecast from her home on a single band radio for her husband, fisherman Oscar Dyson. Before long, she was broadcasting to the local weather conditions to the entire fleet, from Southeast Alaska to St. Matthew Island, even relaying birth announcements, Super Bowl scores, and results of presidential elections. And she did this for over 25 years. Our M2 mooring site, deployed in 1995, honors Peggy's legacy, continuing to provide a voice of the North Pacific. The data collected contributes to one of the longest running time series of its kind. And capitalizing on this 30-year time series, this is the site where I focused my research, which is looking at long-term changes in copepod size in response to warming and cold conditions in the Bering Sea. What I love most about participating in mooring cruises is while our main objective is to deploy and recover moorings, we can only do this during the day when we can see. And the Dyson conducts 24-hour operations. And that means every night when zooplankton perform the largest migration on Earth from the ocean's depths to the sea surface in order to feed on phytoplankton and other zooplankton, I'm there to catch them. I'm often asked what it's like working on a boat in the Bering Sea. So I'm here to tell you, I work about 12-hour shifts, typically from 6 p.m. to 6 a.m. every night for weeks at a time. Research crews can last anywhere from 10 to 30 days, and over the last 5 years, I have covered over 7,000 nautical miles, even crossing the Arctic Circle on my second attempt. But that's another story that I'll tell you if you catch me around town. I, along with the ship's amazing crew, deploy nets attached to frames we call bongos, yes, like the drums, in order to collect planktons of various size. While I'm personally interested in the zooplankton, our bongos capture phytoplankton as well as ichthyoplankton, or larval fish. With one cast, we sample the entire water column, deploying our nets down to 5 meters off the bottom of the seafloor, and then we retrieve them, rinse them down, and our samples are funneled into cod ends, which are these little red cylinders you see here. We can detach these cod ends from the nets, transfer the samples into jars, and preserve them to be examined by professional taxonomists, such as myself back at the lab. Once samples are processed, they're archived at NOAA's Western Regional Center, which is located in Seattle, Washington. And utilizing this assemblage of specimen we've gathered over the years, I've worked with a developer to create an application that can extract size from images collected at the microscope. So over the past 3 years, I have photographed and measured over 10,000 individual copepods that were collected over a span of 19 years. It's one of my biggest achievements. I'm pretty proud of it. So for this study, I had a few hypotheses. First, I theorized that, like those phytoplankton I mentioned earlier, as temperature shifts from cold to warm conditions, that there would be an overall decrease in the individual size of copepods. Among 3 different species of large copepods that I looked at, I found that some showed no difference in size, while others were actually larger during warm conditions. When I looked at small species of copepods, I found that some were larger, some were smaller, and some showed no difference in size at all. Each species had a different response to changes in temperature, and even within the same species, at different stages of their life, I observed that different responses to changing temperatures, which means that my first hypothesis was rejected. I have a couple more. For my second hypothesis, I grouped all the life stages together, and I just looked at each unique species. I was like, this is going to do it for me. Large copepods that undergo diapause, that I mentioned earlier, had split reviews. Some displayed no difference in size, while others were larger in warm conditions. Smaller copepods that forego diapause, they kind of live hand to mouth all the time, had similar responses, which is the opposite of what I expected. All right, so third hypothesis. I looked at the community as a whole. I was like, surely this is the one. This is the one where I'm going to see a trend. And so I expected to see a trend in my data and found none. So I want to take a second to acknowledge that in science, you have to become very comfortable with rejection. Right or wrong, I'm contributing important data that helps inform a community of scientists working together in order to understand the intricate dynamics that make up an ecosystem. So, yeah, it's a work in progress, and I'm really, it's working, it's doing something. So I'm gonna get there. What is my message? Well, I observed a few things while teasing apart my results. Initially, it seems like during warm periods, the conditions are actually favorable for species that rely on copepods as prey. But I want to point out that there's still a lot that we don't know. I do know that we're seeing a shift in species distribution, and warm conditions are leading to an increase in growth weight and abundances as well. There's just more, they're growing faster, and there's more present in that one specific location during the spring, which is translating to an overall increase in biomass availability for the upper trophic levels. But again, I only looked at one time of the year. This is only in spring. That kind of means that shifts in distribution and phenology can really throw a wrench into things come fall, as there is a potential for predator-prey mismatches. The size of my plankton might not be the right size for the fish that are looking for them. What if my plankton are too small and my fish are pretty big and they're looking to put on some meat? They might have to eat a lot more plankton to make up for what they're used to seeing in the fall. Or vice versa. What if they're too big and my fish are too small and they just physically can't eat how big my copepods are? What if they grow way too fast? There's just a lot of questions. So now that we kind of have an idea of what's going on with zooplankton, what about the fish that eat them? This is my colleague, Dr. Lauren Rogers, who is a fisheries biologist. She studies the early life history of larval fish and is especially interested in how fish and their ecosystems are affected by changes in climate. When I asked her what scientists have been observing with regards to pollock, she told me that in the Gulf of Alaska, when it's warmer, pollock are spawning earlier. And they've discovered this by looking at the ear bones or the otoliths of fish, of larval fish, teeny tiny. Yeah, so it's wild. It can tell scientists exactly how old they are and therefore which day they hatched. So similar to counting the rings of a tree trunk in order to tell how old it is, you can count the rings in a fish's ear. And I just find that fascinating. In warm years, larval pollock are hatching earlier, which is the result of earlier spawning by their parents. She says that they don't fully know what this means for larval survival, as it depends on how much food is available, when they start feeding, and how many predators are around. But changes in phenology are an important aspect of how ecosystem dynamics can shift as temperatures begin to warm. Next, as we move up the food chain, I want to introduce you to Marty Reedy, a scientist with the U.S. Fish and Wildlife Service who we affectionately refer to as the birder. Do we have any birders in the audience? Oh, I see a couple. Marty, I met on my first cruise with the Alaska Fisheries Science Center, and during the pandemic, I've since sailed with him from the Gulf of Alaska all the way through to the Beaufort Sea. He is an amazing photographer and very skilled at identifying birds on a rolling vessel, which is no small feat. I fondly recall my first interaction with Marty. I woke up one afternoon for lunch, and I looked out the window and saw what looked like to be snow falling outside my window, but something just wasn't quite right. For starters, the sun was out, and it was falling really slow. Than what I thought. But I just woke up. I've been working night shift, so maybe I was going crazy. Later, I heard that an eagle had caught a bird midair, landed on our boat, and proceeded to consume its prey right in front of Marty and his camera. The deck was covered in feathers for days following this event. And Marty caught the whole thing. The next time I saw him, he enthusiastically showed me the photos he captured and detailed the whole gruesome event for me. Over the years, I have learned a lot about birds in Alaska from Marty. The bird Marty is most interested in observing while he's out at sea with us is the critically endangered short-tailed albatross. They have a wingspan of nearly 8 feet, and mated pairs breed for life, producing only 1 egg a year. Their nesting grounds are comprised of 3 volcanic islands located in southeast Japan, and they eat mainly squid, fish, flying fish eggs, and crustaceans like plankton. Satellite tracking studies have revealed that they leave their breeding island during the summer and move north towards the Aleutian Islands. Short-tailed albatross were hunted to near extinction for its feathers up until the 1940s. Fortunately, a small number of non-breeding birds remained at sea during this period, and that's because immature birds, before they're mated for life, wander at sea for 4 to 9 years. So come the 1950s, a few nests were discovered on Torishima, Japan, after it was recolonized by those young birds who were now sexually mature enough to reproduce. Continuing threats to the species recovery are at the forefront of scientists' mind. Today, over 80% of the population currently nests on Torishima, Japan, which is a 2-kilometer diameter island with an active volcano that's erupted 5 times in the past 100 years. Not great for housing. Between 2008 and 2012, scientists began to translocate birds to entirely new islands in Japan, successfully expanding their breeding colony range. And recently, there's even been successful breeding on Midway Atoll in Hawaii. These efforts have contributed significantly to the species' robust recovery, and population estimates by NOAA is at 10,000 individuals worldwide as of March of 2024, which is pretty remarkable. It's still unknown what effects climate change might have on these birds. Their population rise could mask the other factors that may be affecting them. Warming waters in Alaska's large marine ecosystems may contribute to further expansion of their range and foraging area. There have been 2 sightings, potentially of the same bird, north of the Bering Strait, as well as a northward shift with 2 other species of North Pacific albatross, the Laysan albatross and the black-footed albatross. So, of these 3 ecological responses to warming, this species in time may exhibit a shift in distribution. Now, the last person I want to introduce you to is one of my very best friends, Jessica Krantz, who I also met on my first research cruise in Alaska. She was my bunkmate. And the second I found out that she was the marine mammal specialist on board, I bombarded her with hundreds of questions about whales over the next 2 weeks. I'm sure she loved it. She welcomed every single one of my questions, and since meeting Jessica, I have been embraced by her group at the Marine Mammal Lab, who affectionately refer to me as a fellow whale groupie, which is a title I wear with pride. For her graduate degree, she studied the different dialects of killer whales, which means exactly what you think it does. She literally speaks whale. And guess what? She taught me, and today I'm going to teach you. Right whales get their name from whalers who considered them the right whale to hunt. This is the North Pacific right whale, which is not just one of the rarest species of all large whales, they're also among the rarest of all marine mammal species. And Jessica holds the title for being the person with the most sightings of this critically endangered species. There are likely fewer than 500 North Pacific right whales remaining, while the eastern North Pacific population, which is observed in Alaskan waters, is estimated to be comprised of just 30 individuals. When Jessica wasn't working with the mooring team, she spent her time deploying instruments called sonobuoys, which are short-term floating microphones, which allow scientists to search for species of interest in real time while at sea, regardless of the time of day or weather conditions. She would set the sonobuoy to a specific radio frequency, tossed it overboard, which is very difficult. I've made a mess of myself trying to try it myself. And she used a the radio frequency, she tossed it overboard, it transmitted, and she used a very big antenna that she mounted herself on the flying deck of the Dyson, and she could listen intently for the very distinct call of the North Pacific right whale. And in my free time, I was right there beside her, every chance I got. And I remember one day, Jess had to be in 2 places at once, and I knew it was my time to shine. I assured her I was ready to be a marine mammal acoustician, regardless of my background in zooplankton taxonomy. And I offered to listen for right whales while she tackled another task. So she mimicked the sound of a right whale for me, and I mimicked it back, again and again, until I nailed it. I put her headphones on, and I sat on the floor of that bridge, and I listened for about 20 minutes until she got back. It was the best day of my life. So are you guys ready to learn how to speak whale? I need some audience participation. All right, I'm going to do it, and you're going to mimic it back, okay? All right, so it sounds like a whooping sound, and then it's followed by a gunshot sound, okay? So this is how it goes. I'll do it twice, and then it's your turn. It goes whoop, just like that. Always together. Whoop, yeah. Now we all speak whale. Tell your friends. Currently, there are 2 established right whale critical habitats. One is in the southeastern Bering Sea, and where Peggy's deployed, and the other is just off of Kodiak, Alaska. There are long-term passive acoustic recorders that are deployed in the Bering Sea around the Aleutian Islands as well as the Gulf of Alaska. And the Marine Mammal Laboratory used the data recorded to study the migratory patterns and distribution shifts of the species over time. They've heard right whales on all of their Aleutian Island and Gulf of Alaska recorders, suggesting that both areas are contemporary feeding grounds. In the Bering Sea, they've been consistently heard in the southeastern Bering Sea, but recently they started to hear whales further north, since 2016 to be specific, which they have guessed could be due to prey shifts with changing climate. Right whales are a type of baleen whale, and species of Calanus, which are pictured here, are a critical dietary component for them. So this has actually led to a lot of collaboration between the Marine Mammal Laboratory and NOAA's Alaska Fisheries Science Center to kind of track the migration patterns of their prey, which are Calanus, and see if we can couple it with the acoustic data from all of the moorings, and they're using modeling to kind of predict what's going to happen with these shifts. So, some pretty cool stuff. Okay, let us see. We're almost done. I went by this pretty fast. We're doing great. Okay, so summarizing on what we've learned when it comes to ecological responses to warming. We saw changes in phenology for both copepods and larval pollock, and a shift in distribution for species of copepod, the short-tailed albatross and the North Pacific right whale. They're shifting their distributions. As for the third response, the jury may still be out when it comes to a decline in body size as it relates to my study, but that can always change with more data. So, keep in mind, there are multiple studies that are going on all the time. My specific study was one specific time of year and one location over a long time series. It's really, really hard to kind of track changes when you're isolated to one location and one time of year. Things are constantly moving, everyone's interacting with people, and there's a lot of changes that are going on in the ecosystem. We have changes in sea ice coverage, which are affecting the winds that are being prominent in the area, how much interaction we have with wind with sea surface, which changes how the flow of things work. For plankton, they're drifting animals, so they're kind of led by the flow of things. It's a very, very complex system, and I'm just a small piece of this very big puzzle. There are hundreds, if not thousands, of scientists out there working all the time to understand their specific piece of the puzzle, and then they're collaborating with as many people as possible to share this information so that people with the expertise can put these things together. There are citizen scientists, there are emerging students, we've got people out here that are working on the ecosystem. You'll hear about these people, all these research and different focuses with our speakers today. We have indigenous communities that are providing important information. Everyone is working together in order to navigate change and identify what's happening. We have our finger on the pulse in Alaska, in my personal opinion, and I think we are ready with all of our long-term monitoring to detect changes the second that they happen. To be able to predict what's going on, we're watching really closely. So I feel very confident that not only are scientists navigating change and watching very intently what's going on, taking copious amounts of notes, But the ecosystem is doing the same thing. We're all moving, we're all navigating change, and we're all shifting our boundaries. And I'm really, really excited to show you how one little piece of a puzzle can get put together, especially with the next talk that's coming up. Yvette will add on to how some people's small contributions combined with many others really magnify up into large-scale understandings of what's going on in our ecosystems. With that, I'd like to say thank you. Here's a photo of my spring larval cruise. This one didn't sail very good, but we got a good group photo, and there's just a lot of people that are collaborating together, science crew. It's just been an amazing experience working here in the Alaska Fisheries Science Center, and I just want to thank you for your time.
All right, so we're going to have a few minutes for questions here, and we'll have a couple of mics going around so that you can speak into them so everybody can hear you.
Don't be shy, we speak whale. I think I saw one go up in the corner.
Thanks.
You guys got it.
Nice talk. I was wondering about the composition of the copepods, if you took a
Yes.
look at that.
I did look at the composition of the copepods. I was trying to do a nice little blend between storytelling and science, and thank you for asking that question. We are looking at a lot of changing things. When it comes to the I looked at 6 to 7 different species. Half of them were kind of big. They're known for being big species. Half of them were small. And I made them consistent over time. But sometimes they just don't do so well, and they're just not present at that time in that location, so they weren't present, and sometimes there were more. When I looked at abundances, I kind of really thought that we would have more small guys. I was talking about that borealization, small warm water species coming up into the north. And while I did see that, I also saw an increase in abundance of the large copepods. So that really kind of threw me. We have more large copepods and more small copepods, and they're both bigger. And it kind of just really kind of debunked what I was thinking, but I realized again that there are multiple things happening. And so I kind of think that I'm holding on to hope that I'm still right on my hypothesis, but that there are other things that are happening that are kind of skewing it or masking what's actually happening. So yeah, there are some shifts in the population. Some years where they're more warm, we have a dominance of one particular species, and in other years, we don't see that species. There are some winners and losers, but it's always changing, and it's kind of hard to predict. I had a time series of 19 years, but I looked at every other year. So I really think that my time series just wasn't really big enough or long enough to really kind of get to the heart of the species composition in that one specific place. But it did kind of, lead into some more, questions. He's like, well, what's really going on? So there was differences in composition, and I did look at a variety of species, but some species are kind of hard to differentiate. So I really think that new emerging technologies like eDNA is really going to help when it comes to that composition question.
Very similar question.
Oh, okay.
Yes. Great.
What is the most interesting experience you've had aboard the Oscar Dyson?
Ooh, the most interesting experience I've heard. Okay, let's see. I have a lot. It's really hard to. Interesting experience. I would say. It's kind of realizing that science is complicated and there's a lot of moving parts and things don't always go the way that you expect them to. I hinted to the fact that I made a second attempt to get to the Arctic, and so I'll elaborate that on. I think that one's probably the most interesting experience I had. We were transiting north to the Arctic Circle, and our goal was just from Dutch Harbor, just go straight to the Arctic. Once we got there, then on our way back to the Aleutian Islands, we would sample along the way. So we gunned it all the way to the Arctic, and everyone was having fun. We were getting ready to do our crossing ceremony, and we were playing all these games, and we were having a great time. And we made offerings to King Neptune, and we recited poems to recite at this thing. It was just a wonderful experience with all these different scientists. And then something happened, and the boat started shifting a funny way, and we found out soon that we'd lost one of our engines about 2 hours south of the Arctic Circle, and a big storm was coming, and we had to limp back to Nome. And I was really shocked because everyone was really calm, and I was just like, oh, it's just sad. And then we got back, everything was fine, and a couple years later, we fondly, reminisce about those times that we almost made it somewhere. And I remember somebody saying, like, yeah, that was really scary. And I was like, what do you mean it was scary? They're like, yeah, we were like really, really serious. We were really focused on what we were doing. Like, it wasn't a drill. Like, this was actually real happening. It was a little dangerous. But everyone was so cool-headed and so tuned in to what their job was that they kind of isolated the scientists a little bit, in the sense that all we had to worry about was doing our own job. They're like, we've got our job, you've got your job. And so it was really nice to see, like, how everyone works together, but also when things go wrong or when things change and there are unexpected upsets, how everyone knows how to maintain a good environment and also make sure that they do their job in a safe way. Because I had no idea that there were any problems. I was like, oh, I guess we're just going home. They're like, oh, it was a little serious, but it's okay. I think that that's probably one of my most interesting experiences on the Dyson, is limping back home on one engine.
This may be off to the margin of your expertise, but you mentioned two things that I'm really curious about. One of it is there any nation still killing right whales?
Is the what nation?
Is there any nation still killing right whales?
I do not know that. I don't, I know the United States isn't. And I do know that the Marine Mammal Laboratory works with Japanese fleets to research the whales. They do not hunt them with NOAA scientists. So I really don't know the answer to that question, but I sure hope not, because it's pretty dire for them right now, and they could use a win.
And a similar question, one of the things it's really bad to be is a beautiful bird.
Right.
That never works out well. Why and who was hunting the short-tailed albatross?
Yeah, that's another great question. I don't know who was hunting the albatross. I just know that it was happening. I asked Marty just to describe what he's seen changing, and kind of left out like the details of like how bad it got and why and all those details. So I do know that there are amazing resources that NOAA have for both of these species regarding their history and how they got to where they are. So I didn't do enough research to be able to answer those questions, but I do know that there are great resources available. So sorry I couldn't answer that.
Yes, I was wondering, we've heard or I've read that up there they have amphipods that are like 2 feet or more thick along the bottom of the ocean.
Yes.
And that's where our gray whales go up there and scoop them up. And so I just wanted to know about when you did your plankton tows, or you collected your plankton, so you're doing these vertical tows from the whole water column up. Did you have an opportunity to let it soak for a while to see what would come in? And I just want to know did you see a bunch of amphipods?
Yeah, I love amphipods. I think the craziest thing is, I don't know if any of you guys are familiar with the movie Alien, but that alien was, like, the ferociousness of that alien, it was modeled after the amphipod. Those things are crazy. But our bongo toes are actually oblique toes. They kind of get towed at a 45-degree angle, and so they only get the water column. But there is a different type of scientific method called a benthic grab, which will go down and capture the sediment and get a lot of things. So yes, we do get amphipods in the water column in our bongo toes, but we also would see those that you're specifically talking about in our benthic grab. So two different specialties, benthic, scientists, water column scientists, we're both looking at them.
Thank you. Thank you so much for your talk. Really interesting. Just a question about salmon.
Yes.
Because you talked about the spring biomass being so strong. Is anybody looking at the effects of that on salmon in the Bering Sea?
That's a great question. I think that we might have a speaker. I couldn't say for sure, but I think that there's a scientist here that might have the answer to that question. I am solely a plankton person, and just through rubbing elbows while collaborating on boats is when I learned these random cool little facts that just stick with me. And so I don't know how the spring bloom is affecting salmon spawning and how salmon are accessing food in the water, but I know that there's a salmon scientist who does have the answer to that question. So I hope that my research has a little bell that goes off in their brain and they go, oh, wait a minute, maybe I should shift my focus on this timing and what's going on in this area based off of my research. So that's my goal. Thank you.
I wanted to start off by saying that I appreciate you including people that were involved in your research. I found that personally engaging during the presentation. My question was more in the nitty-gritties of the research that you did on copepods. You talked about a decrease in the mean size of individual specimens that you were studying. I was wondering, was it a proportional decrease in like the entire body or the being of the copepod? Was it specifically in the organs maybe, or organelles?
That's a great question. As zooplankton taxonomists, when we look at size, we have to stay consistent with how everyone's always looked at size over an entirely long time series. And so historically, everyone's always looked at prosome length, which is just the body of the zooplankton, of the copepod, excluding its tail. So my application had to be able to see the whole copepod and then say, that's the tail, that's the leg, that's the antenna. We just want the core part of the body. And then there are these math equations that we have access to that other scientists have, I don't know how they come up with these things, but we can say, okay, my prosome length is this big, and there's this math equation, we can insert it, and we can predict how much lipid availability or biomass is available based off of how big the prosome length is. So that was what I did. I looked at prosome length, and I translated it to biomass. And there were some species and some life stages that were smaller, some were bigger, some didn't change. But like ecology doesn't really, like the ecosystem doesn't care about statistical significance and specific p-values that I set for myself. So I do think that there are some changes we're seeing in the beginning. I think it's really early on, and I think if we keep watching, we might see more changes in the future. But I do think that there's something that's going on there. I hope that answers your question.
Hi, I'm curious where you're planning to go with your research next, now that you have to kind of regroup and form a new hypothesis.
You're gonna make me cry. This is my first job out of college, as you saw, and I've been here for 5 years. And when I was approached to give this talk at Sitka, I still worked at the Alaska Fisheries Science Center, but since then, I have actually got a new job, and I'm moving to a different ecosystem. And so I'm really, really sad. This is kind of like my last big talk, focusing on Alaska's ecosystem. Maybe for a year or two, hopefully I get to come back to Alaska, but I'm completely shifting my focus on phytoplankton. So as you notice, this says planktologist. I am officially a plankton ecologist and a zooplankton, a phytoplankton and zooplankton textile. So I'm the whole base of the food chain, and that's where I'm going. I really focus on it. I talked a little bit about size because I think it's what's kind of applying to what's going on in the area right now. But I actually specialize in a lot of imaging, AI, and machine learning systems. So I have cameras that I deploy in the water that are microscopic, that take pictures, and then I create these training libraries, and I train algorithms to do my job for me. So that translated over really well to phytoplankton and harmful algal blooms. And so now I'm working at the North Pacific Science Center, focusing on harmful algal blooms with phytoplankton using these AI and imaging technologies that I learned about at the Alaska Fisheries Science Center.
All right. Well, thank you so much to everyone for your questions, and thank you, Deanna. So great.